Hello and welcome back. Happy Women in Science Day. Today's webinar, Strategic Implementation of Model-Informed Drug Development for Regulatory Success, is presented by Jill Fiedler-Kelly, President and co-founder of the Cognigen division of Simulations Plus, and moderated by Vice President of Consulting Services, Kevin Dykstra. Before we dive in, two quick housekeeping notes. One, we take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. Second, you may ask questions via the question panel on your dashboard at any time. The Q&A session with all speakers will follow the presentation. Now, as our speakers grab their last sips of water, please tell us a little bit about yourself. Which sector best describes you? We'll give you just a few seconds. We're going to go ahead and close the poll now and see who's in the audience today. All right, fantastic. Well, thank you again for joining us, and it is my pleasure to turn things over to Kevin.
Good morning, everyone. It is my great pleasure to introduce Jill Fiedler-Kelly, who is my boss and also the president of Cognigen Corporation. A little history. She completed her undergraduate in statistics from the University of Buffalo in 1992. And shortly after that, she, along with her two partners, Ted Grasella and Sidi Wallawander, founded Pharmaceutical Outcomes Research. While growing and building that company, Jill completed her master's in applied and mathematical statistics from the Rochester Institute of Technology. Over the past nearly 30 years, pharmaceutical outcomes research grew and evolved, becoming Cognigen in 2000 and part of Simulations Plus in 2014. The company's based in Buffalo, New York, but has contributors that are pretty well spread out across the United States and also in Europe, and currently employs over 50 scientists, programmers, software developers, and other support staff. We provide consulting services to the pharmaceutical and biotechnology companies around the world. In addition to her responsibilities with Cognigen, Jill has been adjunct professor of pharmaceutical sciences at University of Buffalo since 2002, where she continues to enjoy teaching workshops along with a graduate course in population PK/PD. She's published numerous scientific papers and has co-written a book with Joel Owen entitled Introduction to Population PK/PD Analysis with Nonlinear Mixed Effects Models. She's active in a number of professional organizations and has held leadership positions with the International Society of Pharmacometrics, American Society of Clinical Pharmacology and Therapeutics, and the American Association of Pharmaceutical Sciences. She currently serves on the editorial board of the Journal of Pharmacokinetics and Pharmacodynamics, and recently finished her term serving on the board of directors with ASCPT. Along the way, Jill and her husband, Tad, have managed to start and raise a family. Her daughter, Brenna, is a senior at Purdue in materials engineering, while her son, Finn, is just trying to start his college career at SUNY Oneonta in the middle of the pandemic. So without further ado, here's Jill.
Awesome. Okay, thank you very much, Kevin. Appreciate the introduction. Assuming everybody can hear me and things are working well. I'm very happy to be here today to present our webinar entitled Strategic Implementation of Model-Informed Drug Development for Regulatory Success. And before I get into that too far, I'm just going to start with a quick disclaimer, much like our regulatory colleagues would typically include. I'll note that my views do not reflect those of any particular regulatory agency. My recommendations today are offered purely from the perspective of someone who's worked in this field for a number of years and has tried to reflect on what I've seen work well and work not so well. So with that, I'll get going. Okay, so my overview today is that I plan to share some experiences that I've had and thoughts on the effective implementation of model-informed drug development approaches, outline some considerations for optimizing the likelihood of regulatory success with these methods, and finally, as noted in the introduction, if we have a few minutes at the end, we can chat a bit about the significance of today, International Day celebrating women and girls in science. So thinking back on my career, when I got started in the early 1990s, we talked a lot about population PK and population PK/PD, but we really didn't use the term pharmacometrics very much. At least I didn't recall it being used pretty regularly until many years later. So lo and behold, not that long ago, in referencing a journal article that was actually from the mid or late 1980s, I can't remember which, I saw the term pharmacometrics used as a header above the title of the journal article. And that led me to go back and think, wow, I didn't realize, we were talking about it back then. And I looked for some origin of the term, and I found this brief article in JPB, so the Journal of Pharmacokinetics and Biopharmaceutics, which you'll, hopefully recognize as the predecessor to the Journal of PK/PD today. And it was from 1982, and it was recognizing or introducing a new section to the journal. So there was this brief, intro written by Les Bennett and Malcolm Rowland that talked about the use of that term. So, even though that term has really been out there for longer than I was thinking it had, and longer than you might have thought, it wasn't until around the year 2000 that we started to see some academic programs cropping up in pharmacometrics, starting with the University of Buffalo's master's program, and now well over 20 different universities that offer curriculum and degree programs, both masters and PhDs in pharmacometrics. In the regulatory arena, there were many proponents and advocates, even back in those early days, for the use of modeling and simulation at both at FDA and in Europe. And it wasn't really until 2007 that the FDA established their formal division of pharmacometrics within the agency. So today in Europe, there's a EMA modeling and simulation working group. The PMDA holds public workshops on pharmacometrics related topics. And there are these quarterly, at least, pharmacometrics modeling and simulation cluster teleconferences that bring together regulators from EMA, from FDA, from PMDA, as well as Health Canada. And I'm going to come back to that a little later in the presentation and talk about those again. So putting pharmacometrics into context in drug development, initially, we talked about pharmacometric methods, and we used to talk about model-based drug development. And then that terminology was kind of clarified and updated a little bit to better reflect how we really use these modeling approaches. And the terminology was renamed model-informed drug development. And then most recently, model-informed drug discovery and development, or for short, MIDD3. So each of these sort of changes in terminology that have happened over the years, recognizes that expansion in our thinking with respect to the application and the impact of our pharmacometric models. So we really have moved from a way that initially was to learn something from sparse blood samples that were collected either during routine practice or in phase 3 trials. And that really was the origin of the population-based approach. That's what we were trying to address. And now these methods are applied with, to a much broader landscape of using models and quantitative approaches to really inform decision-making, a multitude of decisions, all the way from discovery through development. So today, when we speak about pharmacometrics, or MIDD3, we are often referring to this continuum of quantitative methods that includes the population-based modeling that we started with, but it also includes other approaches like PBPK and QSP and QST. So at the same time that this terminology was kind of changing throughout the years, much more was being accomplished. There were a lot of success stories of using model-based methods that were being celebrated in presentations and in publications. Now recognize that most of those early stories were actually focused on the use of modeling to kill a drug, or to kill a drug sooner than would otherwise, have been accomplished without the use of modeling. And although there weren't as many stories in those early days, again, about modeling being instrumental in the success or approval of a drug, they were happening. And fortunately for us, FDA reviewers have been and continue to really be quite prolific. They've written numerous papers over the years, sharing information about how modeling and simulation has been used in regulatory decision-making, summarizing reviews over different periods of time, looking for wins and success factors and commonality of approaches, as they relate to different decisions. So I think those papers have really been critical in educating the community and decision makers about what's possible with modeling and simulation. And of course, methodological research has continued over this whole time in academia and industry and regulatory, with, in some cases, so many papers coming out of some of these labs that it's actually really hard to keep up with the new discoveries and advances that are coming out. So very, very vibrant area of research. In addition, importantly, there have been a lot of efforts at standardization of methods, which is something that's really important in the context of what we're talking about today. The International Society of Pharmacometrics has formed multiple committees that have investigated issues like model evaluation and data standards and also drug-drug interactions. And there have been ASCPT network and community initiatives on the influence and impact of these methods. And from these efforts have come some white papers and some best practice guidelines around things like exposure response and MIDD3. I'm going to talk about those as we go as well. FDA has also held workshops around best practices, with a fairly recent one in the fall of 2019 specifically about PBPK. In 2014, the Modeling and Simulation Working Group of EMA, which is a group that was formed to give feedback on some of the technical issues around how companies propose to use modeling and simulation in regulatory dossiers, that group got together, they published from their work an activity report where they included this graphic that illustrates a vision for modeling and simulation in drug development. So again, this was 2014. But note, they were working from MIDD, Model-Informed Drug Development, really as a starting point for this, and then working through a whole series of kind of evolutionary steps that involved improving and better informing benefit risk assessment and labeling, and then ultimately getting to improved healthcare as a result of using these methods. So they talked in the report about using modeling and simulation as a platform for a systematic quantitative approach that would support better decision-making for drug developers, regulators, and prescribers. And they talked about the role of sort of open science in this evolution and how important that was as well. So with this as a backdrop in terms of where this all could go or might go, for those in the audience who might be less familiar with implementing these model-informed approaches and maybe have never done this before, I'm just going to back up for a minute and kind of speak to some of the specifics of what we gain with these approaches before we get into the details of how to do it, in a better way maybe, or in a way that helps us with the regulatory implementation. So when we think about these population-based models, population PK, population PK/PD, and exposure response analyses, we can list a whole variety of questions that we answer with the models that we develop. Starting out, there's PK-related questions. Simple things, whether the drug exhibits dose proportionality, if the PK changes over time. Importantly, whether there are intrinsic or extrinsic factors that help to explain variability in PK. Those are the covariate analyses that we talk about very frequently. There's PK/PD-related questions that we can answer with the models we develop, generally around the presence of relationships between exposure and response, the determinants of safety and efficacy, so the covariates that affect PK but also PD, and then how we might characterize that risk-benefit relationship for the compound. And when we use these models, PK and PK/PD, to perform simulations, we can start to address these kind of what-if scenarios that help us answer more practical questions about how we best design a trial, about the probability of success for the plans that we have, a trial that we might already have scoped out, and questions about whether a dose or regimen may be different from the ones we've already tested, might be acceptable or perhaps even better than the ones that we've tried. So this simple cartoon illustrates that benefit risk characterization as well as a way to use our models to help with decision making. So just to walk you through this really quickly, in this slide we see a frequency distribution of individual exposure estimates in our population after administration of a particular dose. And then overlaid on this exposure distribution and relative to exposure on the x-axis here are two curves. The green one that you see up here is going to represent the exposure response model, maybe for the probability over here on the axis of achieving our efficacy endpoint. So the green curve has to do with how we're doing in terms of efficacy related to exposure. And then the red curve down here is representing the probability of experiencing, say, a particular adverse event, maybe as our safety endpoint. So if this were representative of the recommended dose, we'd probably be pretty happy with where that distribution falls relative to these two curves, in that we have an expectation for most of the patients in the population with these exposure estimates of a really good sense of efficacy and a really low probability of the toxicity that we're concerned about in this case. But frequently, we're more in this situation, right, where we observe a good deal of variability in PK that may result in an exposure distribution that looks more like this one, the yellow curve, whereby there's a portion of the population whose exposures are related to a pretty good likelihood of experiencing insufficient efficacy on this end, and a portion of the population that has exposures that are likely to put them at higher risk of observing that particular AE that we're concerned about. So it becomes a question of really what's acceptable. And if we understood better the determinants of PK and PK/PD variability, we might be able to come up with a more optimal dose or regimen or specific dosing recommendations in certain subpopulations that would bring that yellow distribution kind of back to somewhere around where the black one is and feel more comfortable with the dose or complete dosing recommendation. So appreciating that measures of individual specific exposure, which we derive from these population models, give us more information than dose, we then start to think about using these exposure response models to relate those individual estimates of exposure to the individual response that it's experienced. So these individual estimates of exposure, they really could take on a lot of different forms. It could be something like an average drug concentration. It could be sort of a summary measure of exposure like AUC, could be Cmax, could be C-trough, depending on our compound and which one is most related or the best driving factor for that response. But putting this all together allows us to really better understand safety and efficacy. And with our models, we can understand that really both at the patient level, but also we can characterize it at the population level. So the quote at the bottom of the slide, that pertains to the importance of this understanding comes from the FDA's exposure response guidance, which says exposure response information is really at the heart of any determination of safety and effectiveness of drugs. So what I'm referring to in this slide is the paper that I mentioned already about these good practices for exposure response. And this paper really tries to address those good practices more from a practical perspective. And a little later in the presentation, I'll get into some of those more practical recommendations. But at this stage, just to sort of reinforce the ideas that I've touched on already in terms of the applications of these exposure response analyses, I'll just underscore, the usefulness in dosing recommendations is really often what we're most focused on. So with regard to regulatory decisions that can be supported with pharmacometric analyses, this table is adapted from a publication that I cite at the bottom by FDA reviewers that describes how pharmacometric models are useful in supporting a whole variety of different regulatory decisions, kind of grouping them into those that help with approval-related decisions, those that help with labeling, and help with designing trials. So we talk about the specific role of pharmacometric analyses in helping with those particular decisions here. Now, more recently, there's been a focus in some cases on the use of pharmacometric approaches to support the use of a single well-controlled trial by providing that supportive evidence of effectiveness for modeling results. So while I think that certainly can be thought of as a really big accomplishment and success for a modeling effort in terms of the time and cost savings for the program, I'll point out this particular quote that comes from some FDA officials in a more recent article from 2019 that talks about how they discuss that there may be more reasons to conduct more than one trial that go beyond just the regulatory requirement. So, it still may be appropriate to do that. And they maintain that what we should really be concerned about in using model-informed drug development and what we should be shooting for is using these approaches to de-risk both the development and the public health decision-making and ultimately to inform optimal pharmacotherapy. So again, lots of potential great uses. So to come back again to the methods themselves, today when we talk about pharmacometric approaches, there's really several different methodologies that we might be thinking of, and each has its own unique applications, but there's also some overlap between all of them. So to walk you through this slide, starting over here on the right with PBPK modeling. With PBPK approaches, we develop mechanistic models that describe PK in various organs and tissues over time. We rely heavily on drug properties, on known blood flows, and other constants. And we're able to then use these models to predict things like drug-drug interactions and extrapolating and predicting PK profiles in unstudied populations based on the mechanisms that are built into these models. Moving to the top of the slide to discuss QSP and QST modeling a bit, these approaches are also mechanistic in nature. And in addition to PK, they describe the PD effects in organs and tissues over time by characterizing that interaction between the biology of the system and the pharmacology of the compound. So these models then allow us to explore and confirm the mechanisms. They can be instrumental in supporting various drug design questions. And finally, if we move to the bottom of the slide, the population PK and PK/PD modeling that we do is really the most empiric of all these approaches. We fit models to available data, and those models can be more statistical in nature and sometimes are more semi-mechanistic. They can kind of go on a continuum themselves. And they really help us answer questions about characterizing variability in the relationships between dose and concentration and response. So with these population-based models, we can also perform simulations, much as we can with the other types of approaches. But in this case, we're taking into account the expected variability that we've characterized from fitting these models to the data, and we can better understand the impact maybe of different doses and regimens. So again, some commonalities between those approaches, but also some differences. So staying with population-based modeling for just a moment, if we think about how these models can be used throughout a drug development program, the population-based models that we build, we can start at preclinical stage, and the preclinical applications then allow us maybe to bridge into first-in-human dose predictions. Then we characterize the drug's PK with the really rich data that we typically collect from our phase 1 experiments. As we move into phase 2, we typically first start to explore the influence of these intrinsic and extrinsic factors. That's not to say that we can't do that earlier, but maybe we have a bit more heterogeneity in the population now, and we can ask some specific questions about how those factors help to characterize variability. And in some cases, when we plan those things really well, we can actually alleviate or eliminate certain phase 1 studies that we would typically perform by answering the question with our model-based approaches in phase 2. As we move through to phase 2B and phase 3, we can better characterize the time course of response, the variability in response, both safety and efficacy. And we use models to optimize our phase 3 trial design, estimate the probability of success with the designs that we have planned, and ultimately, determine the most appropriate dosing recommendations and support labeling considerations. So to address the use of these approaches in support of regulatory milestones, I think it's really helpful to be cognizant of the many guidance documents that in some way address model-related issues. So this slide is an attempt to list some of the FDA guidance documents with content that I think is really relevant to the issues that we're discussing today. The current population PK guidance at the top here, notably, is in draft form, where it reflects really a major revision to the document that we used for many, many years that was originally published in 1999. The exposure response guidance that I referred to previously is from around 2003. And some of these other guidances really are much more recent, with one that addresses PBPK from 2018 and one on innovative trial designs, which are often supported by simulations. Importantly, the FDA has also initiated a program a couple years ago called the MIDD pilot program, which is a 5-year program that was intended to enable and encourage early discussions between regulators and sponsors on the implementation of modeling and simulation within a program. So on the strategic issues about modeling and simulation. So just to go into that a little bit more, the wording I've included on this slide comes from the FDA's website for the program. Calls out the use of modeling and simulation at the top here to improve trial efficiency, to increase the probability of success, and to optimize dosing in this sort of in silico environment. They describe the issues that they typically focus on in these discussions as being around dose selection or estimation, clinical trial simulations, and predictive or mechanistic safety evaluation. So, with this program, again, if you're, just getting started and thinking about using modeling and simulation in your program and don't have a lot of experience with it, it might be a really good idea to think about this and to, put in a request to get the regulatory agency involved in helping you think about how it can be used. While there's no guarantee that a particular request would be granted, of course, limited bandwidth to accept all of these, in cases where you're planning to use it to support really key decisions or maybe there's some specific challenging issues around your drug or your program, it might be worth it to try and get that input early so you can align on what will be acceptable instead of waiting until you've kind of banked on it and tried to use it and then finding out that, there were some key things that could have been done differently. So it's important that you're aware of that. And then when we think about the product that we are creating from our modeling efforts. So here I think about the product that we're developing as the output from our modeling effort, sort of the knowledge that we have captured that helps us to inform decision making. That's usually put together in some sort of report or something, right, that we produce and send on that goes to the reviewers. So I think when we think about that, it's really helpful to understand the job or task of the reviewer on the other side that is receiving that output that we produce. So to know, what they're facing and what they're up against, this part of the manual of policies and procedures for reviewers in ClinPharm is something called the question-based review. Falling under good review practices, this is a tool that reviewers use in completing their review to help kind of guide the content of that review. It promotes consistency and communication, and helps to integrate the modeling and simulation results into ways of addressing exposure response and posology. So this slide includes some of what are sort of listed as the high-level clinical pharmacology questions that are included for the reviewer to answer or address based on the information that we provide. So I won't read each one in detail, but you'll see by the text that I've highlighted in purple, if you can see the difference here, that there's some common themes that we've been discussing, and they are reflected in these questions. How are we using modeling and simulation to provide that supportive evidence of effectiveness? How are we supporting the dosing regimen and the role that modeling and simulation is playing there? Are alternative dosing regimens appropriate, maybe for subpopulations based on those intrinsic factors or the covariate analysis that we did, or maybe the extrinsic factors? So the QBR tool itself goes on for many, many pages. And it lists a whole bunch of different questions besides those high-level ones that we looked at on the last slide. And the reviewer is basically tasked with answering these questions in their review in order to give a really complete picture of the clinical pharmacology issues. So many of those are really addressed with the model-informed drug development approaches and the modeling reports that we provide. So again, we won't go through every single one of these, but you see how having an understanding of the questions that need to be answered, the content that the reviewer is looking for in our reports, should give us better insight as to how we write them to facilitate the reviewer answering these questions. So, starting out, there's some questions about general drug attributes in clinical pharmacology, characteristics of exposure response, right? We've talked a lot about that already, and QT issues. Under PK, a whole variety of questions that are just characterizing, the processes that we talk about, but also some specific questions here, about single versus multiple dose PK, about addressing the pharmacokinetics in healthy volunteers versus patients, and whether those are different. And then some specific questions about subgroups, or whether the PK is linear or nonlinear, whether or not the PK changes with time. We usually have, start out with our short-term studies and we extend to longer term. So that's something we need to look at and address and think about based on the data that we've collected. And of course, then, if you go feed this back far enough, then these give us good insight into making sure that we're collecting the right data to address these issues as well. But inter and intra-subject variability in PK in healthy volunteers and patients, and then the causes of that variability. So detailed questions going into the covariate analyses that we do about both intrinsic factors and extrinsic factors, which ones matter and influence exposure and response, and then would we make a dosing recommendation that, based on those influences that we find. And then of course, drug-drug interactions as well. So there's just some insight into, I recommend that you look into these specific questions and make sure that whether it be in your modeling report or in other clinical pharmacology study summaries that are produced and kind of go into typically the section 272 of the CTD, that all of these things get addressed in some way in there. So while we've discussed some of the relevant guidances and the other tools for FDA, of course, the EMA has their own series of guidelines as well. And in addition to the guideline documents, like the first one on reporting the results of population PK analyses and reporting on PBPK analyses, They also have what are called these Q&A documents on a website that you can refer to. So some typical questions that are asked frequently and kind of the agency's opinion on those. And then some reflection papers on very specific issues that relate to MIDD kinds of issues. So recognizing that we're typically interested in achieving regulatory success in multiple different regions or globally, we need to understand the differences, right, in the expectations between the different regions. And then, of course, we have to comply with all of those. So the topic of global harmonization of regulatory efforts is talked about pretty frequently. With respect to harmonization between the FDA and EMA in particular, there was a recent review article, Clinical Pharmacology and Therapeutics, and an editorial associated with it that kind of addressed this issue head on in this title of Are Regulators Talking to Each Other Across Borders? So it was really trying to investigate that a little bit and see what the conclusion was. And from the work that they did, the conclusion was that yes, indeed, there is quite a bit of communication that's going on amongst regulators, maybe more than we who are not regulators would appreciate. And there are these clusters, as I mentioned before, that have been established around certain topics within modeling and simulation and pharmacometrics. And then within those clusters, there are groups that are engaging in dialogue really regularly. So it's like kind of at least quarterly, but sometimes more often than that. In some cases, they were talking about communicating almost on a daily basis. So, where does that lead us? Okay, sounds like a great thing, right, for the regulators to be kind of on the same page and talking to each other. But does that mean that the reviewer from FDA, who's getting your modeling report, and the rapporteur and the co-rapporteur from EMA are going to agree on, what they think of your output? And unfortunately, of course, the answer is no, or definitely not necessarily. Okay, it certainly does happen that there is general agreement between those, but not in all cases. I'll come back to that in just a second, but there were these two reviews that were done of FDA and EMA submissions over a specified period of time. And they really tried to look for agreement or differences in outcomes between the US and Europe. And the good news from their work too is that they found considerable agreement really on the ultimate decision regarding the approval of the drug. And there were very few instances of differing opinions, especially when the two agencies were reviewing the same data package or studies. Sometimes those differed because of the timing of the issue, and that was one of the reasons. But when it was the same package, they generally were almost always in agreement. But going back to the modeling questions in particular, our experience, or maybe I should say my personal experience and the things that I have seen over the years, has been that there really can be quite a bit of difference in the questions and the suggestions around modeling that come back from the two regions, as well as from the two rapporteurs in the same region. So while we really try to comply with all the requirements and write as clearly as we can, note that there is still the human element to the review process, which often results in different questions that can arise from just the unique perspectives and background and experiences that the reviewers have. So I'm going to tell my computer not to restart right now. Sorry about that. All right. So for those listening that might not have much experience in implementing modeling in their program, I think one of the key concepts that's important to become comfortable with is this idea that the use of modeling, as I've described, is really evolutionary. There's this seminal paper by Lewis Shiner about learning and confirming in drug development that is really very, very frequently cited. You probably have heard of it. And it's from, I think, the 90s or something. There have also been several modifications over the years to people kind of building on that and thinking about it with sometimes a prediction step frequently included in that as well. But in any case, if we implement modeling early on in a program, we should really have an expectation that the models we build and develop, they change over time. There's not just one model even at the end of the day. There can be many models and different aspects of them, and models that are more appropriate for different situations or data sets or questions that we want to answer. So, that is because we use models to test assumptions and learn things, and then we simulate from those models to make predictions and to design new trials and to collect data, and then we start that process all over again. So these models definitely change over time, and we should expect that, and that's not unexpected at all. So this table is an attempt to characterize some of what I would call maybe typical uses of population-based modeling and simulation throughout a drug development program, where there's many formal iterations of models that expand and change over time, and there's many less formal iterations within each of these steps. So starting out, we develop a population PK model, and maybe in a typical program, we start that, we could start that preclinical, of course, but if I'll just talk about clinical development at first, we might start that after we have a handful of phase 1 studies completed. We pool those studies together, and we start to characterize the PK characteristics of the compound using, again, maybe a population-based approach. And then as the phase 2 studies are coming to completion, we take the data from those studies and we fold it into our data set and let that data set expand and grow. And that enables the model to expand in that now maybe we add the investigation of intrinsic and extrinsic factors, and we look at covariate effects and things like that. So that model changes, and again, it changes when we have phase 3 data. So it went from just characterizing PK to understanding effects of covariates, and eventually generating those individual specific estimates of exposure that we're going to want to relate to our PD endpoints. So there's questions, even at the PK stage, that we can answer with these models, where we're, maybe evaluating the feasibility of, say, a different type of dosing. So maybe our program started out with weight-based dosing, and we want to evaluate the feasibility of fixed dosing. So with our PK model, at the very least, we can use it to simulate those scenarios and to understand the impact on the exposure distribution, and if we're likely to see really vastly different exposures from light patients to heavy patients in that particular example. And then, of course, we can start to develop a plan to evaluate the viability of these alternative dosing strategies, but only when we have a good understanding of the PD impact of that as well and how it relates to exposure response. So we might build PK/PD models initially for a particular biomarker that allows us to confirm the mechanism of action, to understand the impact of exposure on the magnitude of response that we're seeing, which again helps us to think about the dose range we should be in. Our exposure response models, as we get a little further, for the primary and secondary efficacy endpoints and any important adverse events, help us to characterize that relationship, understand the determinants of safety and efficacy. And then when put together with the PK models that we start with, they allow us to simulate probability of success and really think about moving forward with the program and then what doses we should be recommending. As we finish, say in a typical program, we're getting toward a little further on in the adult development program and we understand what's going on there, then we might try to use the model to simulate what dose we would start with in a pediatric population. That's a really common use of modeling and simulation, and maybe other subpopulations or special populations. But with peds, then we might run our first trial, collect a little bit of data, again, refine that model over time as well. So it just gives you an idea of some of these, typical kind of evolutionary uses of modeling and simulation and how things change within a typical program. So on the next 2 slides, I'm going to get to some of that specific feedback that's given in these 2 documents that try to capture the good practices for exposure response and for MIDD3. In part, these ideas behind these manuscripts was to enable greater consistency in the practice, the application, and the documentation of these approaches, not only because we wanted to enforce that within the community, but also in part to provide regulators with a basis for perhaps developing an MIDD3 related guideline someday. So this first slide is about the exposure response good practice manuscript. It was published in 2015, described by the authors as intended to focus on those practical issues of exposure response analyses and really to promote, kind of consistency and standardization across how we use it so that then we can think about how to, maybe even compare analyses for different compounds. So the authors detail a variety of possible questions and question types which might be relevant for ER analyses. And they characterize them in design-related and interpretation-related questions, things like whether ER simulations support the phase 3 dose regimen, whether the ER relationship supports evidence of a treatment effect, things like that. And then they discuss about how those questions really should be linked to the particular analysis that we're going to do. And they do provide in the document, some specific equations for different types of models and how those relate to answering certain questions. They also go through some of the common issues that come up in these analyses and addressed what they feel are the best practices to deal with those issues, including things like pooling the data across different studies, which is often really helpful to give us a broader range of dose and exposure that we can work with in developing our models. How we often need to drop the patients that don't have exposure-related data in these models, but of course we are typically including the placebo group in that mix. We need to choose the appropriate endpoint variable, choose the appropriate exposure measure. That can sometimes be complicated, and we need to think about those different exposure measures like the ones I mentioned earlier, and how they can be interpreted similarly or differently. Like when we have very different dosing regimens, sometimes the definition of a particular exposure measure changes, and so it's harder to use that across different dosing regimens, and others we can use across many. In any case, they talk about how we might need to add a dropout model if we have significant dropout in the model and it matters to the response that we're seeing. They also get to the point where they describe these exposure response analyses as often more supportive evidence rather than the primary analysis. And in part, that's due to this issue that we've been talking about for a while about the possible presence of confounders in these analyses. So we don't design our studies to achieve a certain exposure. And when we look at then the relationship between exposure response, there's this possibility that there are confounding factors in there that kind of muddy the interpretation of that actual relationship. So they talk specifically about some recommendations regarding the inclusion of covariates to address that and ways to visualize the data as well. So definitely recommend taking a look at that paper. The publication addressing good practices for MIDD3 provides recommendations on planning and conduct and documentation of these analyses. And it really begins, and I don't say it right on the slide here, but one of the most important things in this paper, and we'll come back to it on another slide of mine, is that it begins with the development of a strategic plan. So, we establish that plan for how we're going to use modeling and simulation, ideally with multidisciplinary stakeholder input and buy-in. So we want to get all the relevant folks from, if we're doing this in an industry organization, a pharma company or biotech company, we need to get buy-in from folks from a lot of different related areas as to how we intend to use modeling to support these particular arguments. Sometimes it's complementary to other approaches, and sometimes it's the fundamental argument, but it really speaks to making sure your organization is aligned on how you're going to use it. So other key elements of the good practices include a clear statement of the objectives. That's going to come up many, many times. Transparency on the assumptions and their impacts on the eventual findings, a clear description of the findings themselves and the associated recommendations, and then enough details to support the reproducibility of this work. So for documentation purposes, they specifically recommend in the article that if the intended purpose of the modeling exercise is to support internal decision making, that in that case we need an analysis plan, because we should just be doing that. I'll come back to that issue as well. And an abbreviated or sort of a brief report that describes those outcomes. And then if the intended purpose of the modeling effort is to support a regulatory application or a regulatory question, then we need an analysis plan, a simulation plan, and a technical report. Of course, each of these documents, those deliverables that I'm talking about, the analysis plans and the reports, would be prepared in accordance with the recommendations based on the guidelines, and we're going to talk about those in just a minute. But in this paper, there's extensive background information that really talks about the value of MIDD3. But the paper also includes, numerous examples of papers that support different types of modeling applications. And I would really recommend this as a very good reference, especially if you're looking for an example that might be similar to your particular situation. You'll probably be able to find one in that article that kind of gives you a guide to how someone else approached dealing with the same issue using modeling. So given that paper was out there and we were using these methods pretty commonly in industry, in 2017 there was a survey done of industry and regulatory respondents that was kind of a follow-up to that best practices paper, good practices paper, and dealing with how are we doing on using these methods. So the consensus from that survey is that there was a good match, with some gaps it said, but a good match between the practices described in the paper, what was actually happening in industry, how companies were using it, and what the regulatory expectations were. And they also noted there had been a big increase in the degree of impact of MIDD3 on decision-making over the 5 years that predated this particular survey. So other major takeaways from the survey included calls for, the importance of educating decision makers, starting that strategic planning for the use of these model-informed approaches early with input from regulators. Again, getting back to something like the MIDD pilot program, and also the importance of professional organizations in playing a really crucial role to help advance the impact of these methods. So putting this together and thinking about what are some of those essential components to successfully navigate the regulatory hurdles. And this is, again, comes from these articles, but also my personal opinion on what kinds of things you need to have in place and how you actually do this well. So I'd say one of those essential components is learning and awareness, right? We have to understand what the expectations are. We have to understand what is expected of us from the guidelines and the regulatory documents. It might be a good idea to investigate similar recent programs, if there are any, in order to understand what issues were important from a regulatory perspective for those. We need to have a good grasp on the methods themselves that we're going to use in this area. Second component I think that's really important is having an ecosystem in place to facilitate seamlessly implementing these methods. So, I would say this isn't something that you're going to develop overnight and say, okay, we made the decision, we're going to use modeling in our programs, and we're going to use that to inform our decision making and to inform our regulatory decision making as well. This is something that really you need to kind of build up over time. That ecosystem is going to include things like having tools in place that include validated software, canonical documents, which I will come back to and describe in a little bit what I mean by that. Importantly, educating those decision makers and stakeholders within your organization and seeking their involvement and input to how these methods are to be used early. So it is going to be a matter of, trying to describe to them these case studies and the situations where it's been used really well in order to get that buy-in in some cases. You want to have skilled project management support to align the timelines, because you're going to be working across different disciplines for data availability and document reviews, and then putting together the NDA at the end of the day. Having a quality system in place to address the QC and the QA issues and the scientific review. We'll talk about that a little bit more in a bit as well. And then finally, execution is really key. So developing and following that strategic plan that we put in place, the recommendation from the MIDD3 paper, carefully designing the studies to collect the right data in the first place, paying attention to the details along the way. And then I think, underlying or overlying, however you want to think about it, this whole sort of plan needs to be, I think, a willingness to what I'll say is over-communicate. We have to communicate within the team of folks that are working on these efforts and trying to make them work. We have to communicate between the team members, the modeling and simulation or pharmacometrics folks, and the related functions, ClinPharm and all the other areas. We need to communicate with upper management. And of course, we need to communicate with the regulators, as we've talked about. So if you kind of try to just do a modeling exercise in a box and then show everyone later what you did and how you learned things that could be helpful, it's going to be really unlikely that those results are actually going to be used. So only with really consistent communication among the team and all the other folks from beginning to end will you sort of start to gain some momentum from the first time you try to use these methods to the next and get better and better at it, and eventually have it become a really typical and expected part of your program and have the results actually become part of the expected package that's used to make decisions at your company. So as I mentioned on the previous slide, if it starts with that strategic plan at the program level for how we're going to use modeling and simulation, then a level down at the level of the particular modeling exercise or the analysis is the analysis plan that you write. So everybody has a different name for this: modeling analysis plan, just analysis plan, data analysis plan, modeling and simulation plan, whatever you want to call it. It's the document that is really akin to a statistical analysis plan for a traditional statistical analysis. It's basically the document that you write before you do the analysis to keep you honest, right? It's where you pre-specify what you're going to do and how you're going to do it for modeling, how you're going to make decisions and how you're going to choose that one model is better than another and which one is good enough to answer the questions that are of interest. So there's several sources for recommended analysis plan content to walk you through what kinds of things should be included in the plan so that you set yourself up for then performing and executing on that based on these good practices. So I think if you have a really good analysis plan in place and you follow it, you're setting yourself up really well. So what I provided here is a high-level outline of the recommended content based in part on the MIDD 3 good practice paper based on content in our textbook. I won't go through all the details here, but important for an analysis as well as a simulation plan is, again, starting with that clearly stated purpose, the questions to be answered, the objectives, however you want to say that, like what are we trying to do and how are we going to use modeling to address those particular questions. Of course, getting buy-in on the plan itself, this whole document, in particular, those questions to be addressed, the goals and objectives is really important. But the other thing that's really important in the simulation section, I would say, is trying to get alignment on the scenarios that you will simulate. That's going to set you up for success later as well. In our experience, it is that portion of these analyses that can blow up from a resource and planning perspective, right? Once you get folks engaged in what we can do with models and they see what's possible in these simulation scenarios, we can go from having 4 different scenarios maybe planned to suddenly doing 24 different scenarios. And let's try this, and what if this, and what if that? And as we continue to, throw all those things out there and try all these different approaches, we'll find later that some of those, just really did not make sense. Maybe they weren't reasonable to try. So again, thinking really clearly at the beginning and saying, what are the questions we want to address, and outlining those is going to help as well, with respect to your resource planning and your timing and making sure that you can deliver on time. So when it comes to reporting, before we get into, what should be in a report, these are just some general, thoughts and ideas about reporting. So the first, we're basically telling the story. Yeah.
Jill, I'm just going to break in really quickly. We're getting close to time.
Yep.
We can keep going, and so for the folks that have that time, please feel free to stay here. For the folks that have another engagement, please know that these slides and the talk will be recorded and will be available on our website. So don't worry about that. I know that there's a lot of information that you still have, and also some items about the Women in Sciences part. So with that, I'll stop and go forward.
Okay, thanks, Kevin. All right, so I'm going to keep going with this one because I think this slide is important, and then we'll go quickly through the recommendations that are in the guidance, which is what comes next. So I think, remember that, this report that I reference here and have a citation for an article that my colleague Kevin actually was instrumental in putting together based on a survey. The report that you write is both about communicating your results, but it's also about supporting the reproducibility of what you've done. So it reflects how well you can communicate the findings, and telling your story in a very clear, concise way is, of course, very important. But also, I think, providing your rationale and the reasons why you believe or don't believe something, why you followed a certain path and not another, is really critical to someone else reviewing it and understanding your thought process. So one factor, writing in real time might alleviate some of those issues and sort of so you don't have to go back and think about these things later on. But just because you write in real time doesn't mean you actually have to report on every single thing you tried. So I don't want to give you that impression. There's definitely value in being able to kind of step back after you've made it to the final model and see the whole story more clearly and then refine that messaging. But another tip is really trying to get alignment on those messages with your key stakeholders early and as you go. So as you're doing the modeling exercise, if you're keeping folks involved in how you're making those decisions and what you're finding, it'll help to focus your writing and ensure that the modeling report fits in really nicely with the rest of the submission package, because that's really important. So of course, following the recommendations and the guidances is important, but thinking as you're writing that the person reading this is probably thinking why as they're reading it. So, they're not sure of all the decisions you made and what was going on in your head as you were doing it. So keeping in mind the questions that the reviewer needs to answer and making sure you're providing all that information is really critical. So this is another slide that comes from a particular reviewer's presentation at the joint statistical meetings a few years back. And they have some very specific recommendations about what should be documented. There's only a few here that I think are really unique and should be pointed out, just to make sure the folks that are doing these modeling exercises are aware of these requests, if you will, from reviewers in terms of do's and don'ts. With respect to covariates, one of the specific requests is that we stratify our VPC and diagnostic plots by relevant covariates, and that we don't, on this side, describe the covariate effects just in terms of their impact on parameters, but in fact, their impact on exposure and response being really important there. And then when it comes to putting everything together and the electronic files that we submit, making sure that the ID number, if we created a new ID number for modeling in our modeling data set, that can be tracked back to and aligned with the ID numbers that are in the clinical study reports and in those data sets. That becomes really important to the reviewers as well. So again, I'll go through this quickly, but this slide just shows some of that typical content of a modeling report. In this case, there's pretty good agreement between the FDA and the EMA in terms of what should be included in these reports. A couple minor differences about the FDA wanting a few more levels of summary up front, and the EMA is a little more specific about particular graphs and diagnostics that they want to see, but otherwise, you get the idea of what's in these typical reports. Results are pretty straightforward. Discussion, one of the things there is making sure that the information is presented in terms of its consistency with the standalone clin pharm study results. That's important. And then in the appendices that we provide, providing that run record, which is kind of a summary of the different models that we tried and kind of a high level of what we found from those different models. Now with PBPK reports, there's a bit of difference to my read between the FDA recommendations and the EMA recommendations. What you see here for FDA is kind of a typical report format where shoehorned into these typical reporting sections are the things that are very specific to PBPK. So the physical chemical properties going in the introduction, the modeling parameters in methods, things like that. But otherwise, this is kind of maybe more similar to what you would expect or what you would have seen. But the EMA's guideline on reporting of PBPK is actually a little bit more unique and different, I would say, in terms of what the expectations are there and how this the report would come together. And they have specific sections like the qualification of the platform, and they talk about exactly how that would be done in the guideline. So one of the key components I mentioned earlier is having a quality system in place to support all these activities. That has to do with QA and QC, quality assurance, being those activities or processes that support data integrity and reproducibility, which has to do with training and validation of software. Quality control are kind of those activities where we're checking our code and our output and things like that. Because there's so much that we develop and produce for modeling efforts, we really can't check everything. Right? It's just not possible. So we might develop a risk-based approach where the key and most highest impact portions we QC to a greater extent. We can have a certain approach for data, for models, maybe the key models that are involved in decision-making steps, we're going to want to check more carefully. And then with respect to scientific review, we can do that either inside the organization or outside the organization by someone independent of who did the work. There is maybe value going outside the organization for that perspective of someone who wasn't involved in what was going on and giving that kind of input. We have some posters on forensic pharmacometrics that we talk about here. I'm going to skip over the model credibility assessment, but it's kind of a newer idea about how we think about models that is kind of aligned with what the EMA has in their PBPK guidance. Canonical documents, just to come back to that since I didn't define it better before, is this idea that we can have a standardized or templatized version of our report to kind of ensure consistency in the reporting. It helps us make sure we didn't forget something, and it facilitates review, and it facilitates bringing the documents together, and is something that could be incorporated into a reproducible reporting tool as well. So definitely something you want to consider. In this slide, I was just attempting to point out an article that I thought was really nice in 2020, early 2020, that was put out by the editors and associate editors of CPT PSP that asked them to look at what the industry would look like in 2030. So basically to get a view for how we're going to use these models. And they presented a really nice picture of how far we can go with our pharmacometrics approaches in integrating all these tools and having better tools to inform precision dosing. So with that as a possible future state, I hope, I'm sorry I had to go fast at the end there, but I hope that you've taken away some good suggestions for improving the current utilization of model-informed approaches, and in particular, some ways to improve your likelihood of regulatory success. Remembering, a clear statement of questions and purpose should come first. Carefully selecting the optimal approach. Consider setting up an ecosystem to support the implementation of these model-based approaches. Paying attention to the details as we execute along the way, and then when we report, keeping the audience in mind and remembering the questions that they need answered, and then internally making sure that we're communicating really well and seeking out opportunities to communicate with health authorities as well as the folks within our group in order to keep things aligned and have better success. So with that in mind, I will just pause right there and see if we want to do a poll or we want to take some questions before, and if we want to move on to this section of the talk.
Hi, Jill. Great. Yeah, let's take a quick poll before we run through the Q&As. There's lots coming in. Is your organization currently using model-informed drug development strategies? This is a great question to help understand how we might be able to help, or how we can point you to our website for good strategies and feedback. If you haven't already done so, I invite you to send your written questions to the group using your questions pane, and I'll go ahead and turn it back over to Jill.
Okay. So Kevin, I'll go on your recommendation here. Do we want to address any of the questions, or do we want to go on to talking about women in science a little bit?
We have some really great questions, but I would say that that an important part of this talk is also to talk about women and girls in science. So I would say go ahead and do this part.
All right. So really quickly, having worked my entire professional career in pharmacometrics, I came into it, as Kevin said in the intro, with a less traditional background, a master's in statistics. So, one might expect that I would have experienced maybe an even more challenging path in terms of opportunities as a woman in this field. But honestly, on the contrary, I feel pretty blessed to have been generally supported and encouraged along the way. In speaking with a lot of my colleagues at Simulations Plus, they actually had similar experiences of support, which is definitely not meant to diminish experiences, to the contrary, but I think it's a good reflection on our area, with us as a small sample. I think that speaks volumes for this area. So starting out in a research group at UB, there were 3 of us, 1 man and 2 women that started Cognigen in the earliest days. Our staff was very, very female heavy. In those days, we had scientists, statisticians, programmers, and in the very early days, data entry personnel, and they were really predominantly female. Our Cognigen division today is still majority female. 57% of our scientific consultants, 45% of our programming staff, and 100% of our project managers. So I've been in many situations where an entire project team comes together and they are all female. I think, one of the important things today to talk about in light of this topic is, all of us, right, men and women alike, have really been greatly impacted by COVID. And that's both professionally and personally, and in ways that bleed the 2 of those things together more than they once did. So one of the reasons for this day is to recognize and promote women in science, so that we encourage girls and young women to pursue careers in science where there's been this historical underrepresentation. And unfortunately, reading the news these days, right, you hear that there's been a disproportionate impact of COVID on women in the workforce. And so 30% more women have dropped out of or left the workforce since COVID has started. And last month, it was something like 4 times as many women. So it's perhaps getting even worse. And, that's due in part to the sectors that were affected and the predominance of women in those sectors, but also due to the role that women tend to play in caregiving. So, I think it just highlights the uphill challenge that sort of remains in this area and just reinforces the importance of it. So the other thing I just wanted to mention, I think, often when we talk about this topic, we highlight idea of females as role models. And we've come up with some of these, lofty ones that we've seen over the years, and Marie Curie is always mentioned, but, closer to our area, we got folks like Gertrude Elion and the folks that were recognized for gene editing for the Nobel Prize just this year. There's a growing number of female executives in pharma and biotech. And then importantly, right, we have female healthcare workers and teachers and counselors who are risking their own safety. And for me, I would say that most of us in modeling and simulation in this era have been really fortunate that we can do our jobs quite effectively, right, with our computers and for work from home situations. So that's lucky. But I would say from the time I got started in this area, there have been numerous women that I think were truly instrumental in the growth and evolution of our field, several who were already really well established and successful scientists that I looked up to in those early 90s when I was getting started. Sandy Eller-Heiligen at Lilly was doing great things and talking about modeling and its implementation a whole lot. Janet Wade at the regulatory agency in Sweden was one of those folks. Of course, France Montré, with all her publications and research in this area. In academia, Marilyn Morris from UB had a thriving research program and where I first learned pharmacokinetics. And then, over the years, people like Holly Kimco have invested a lot of their time in writing and publishing and education. And I think that's been really important to the area growing. And of course, like Stacy Tannenbaum, who's been really instrumental in establishing the global community that we have today around pharmacometrics and bringing together the separate users groups that we had into, the American community of conference on pharmacometrics, and then eventually the international society that it is today. So areas changing very rapidly. Many of us are working really hard to focus on doing good science, thinking about the growing impact of our work. I would like to recognize all of my female colleagues at Simulations Plus in all of our divisions, especially the scientists, programmers, and developers, who I think really inspire me every day in their dedication and commitment and how they support our clients and work really diligently and are role models for all of us. So I will stop there.
All right, that's a wonderful summary. We actually did get some really great questions. Stacy, who you mentioned, unfortunately, she asked a really good question, but she's left. So with that, she's you snooze, you lose, I guess. Lisa Sheehan had a really nice question, which reads as follows. This is focused on a large development program. However, can you speak about how POPPK modeling, and I would say MIDD approaches generally, can be applied to support a very small, i.e. less than 100 subjects development program? And I would think that might be in the context of a rare disease, maybe rare and chronic disease, but if you can maybe elaborate, that would be great.
Yeah, that's a great question. I think obviously there are some different and unique challenges when we have these really small sample sizes, right? And so some of the statistical and model-based methods that we use need to be a little bit different. But I think it maybe highlights even more in those scenarios where it's particularly challenging to implement these methods and make sure that we're going to get buy-in and appropriate. Everyone will be on the same page with the results that we come up with. It just speaks more to those issues of starting planning early, thinking about what's possible, getting the regulatory input, and within the organization, kind of reaching out and looking for those other examples of programs that have done something similar. I think the general ideas and the overlay of implementing it early and having all your ducks in a row apply to programs big and small, with some extra challenges in those areas. And again, just reinforces the need for that buy-in in my mind.
Can you talk a little bit about leveraging prior information in that kind of a context, sort of looking?
Definitely. Yeah, that's an important point. And something that comes up really frequently when we do pediatric applications, right, is this idea that, maybe we use a Bayesian approach to think about what we know already, whether that's from, in a pediatric situation, whether it's from adults and where we understand the pharmacokinetics really well from bigger populations and bigger sample sizes and more informed data, or in another situation, maybe it's from the literature or from mechanism, based research, where we can use that as prior information and use that to leverage the very small amount of data that we're able to collect in some of these, harder populations to study. And so, statistically, I think that's an important approach that can be applied as a method within a model-informed program.
Great, thank you. We'll get to Stacy's question now, which I think is a really nice one. So she talks about harmonization efforts between the FDA and the other worldwide regulatory bodies, especially EMA. She says one big difference is that FDA has a pharmacometrics group of their own that asks for code and data, and they frequently will do their own analyses. Whereas in the EMA and others, they rely more on the modeling report that the sponsor submits and follow up with technical requests or qualifications. Can you talk about how this difference results in different kinds of questions among, from the global submissions?
Yeah, that's an excellent question, and this has come up before in other forums. I really do think that's, basically at the heart of a lot of the differences that we get back in terms of questions from one, side of the ocean versus the other. I think the ability of the FDA reviewers to take the data sets and the code and to do their own analysis and to play with it and to try things really gives them a much better picture than just counting on that report that we've written and the, whatever code we potentially could include in an appendix or something, but not having the data to actually play with it and either try for reproducibility purposes to justify that, those results are expected and they're what we would get too if we applied the same thing, but then to ask other questions. I think that is at the heart of a lot of those differences. It's not, entirely the issue, but I do think that's a big part of it. And I think the FDA reviewers' willingness to get in there and play and try other things really gives them a very good picture. And we're all, maybe not everyone on this call, but those of us who are modelers and scientists, you're going to have a different take on something than someone, sitting in the office right next to you would. And so the ability to investigate it a little bit and poke at it really gets you to a place where you've got more confidence in those results, perhaps, or you can ask a different question than you could if you're just looking at that modeling report, and it becomes so important that the report is well characterized and describes the rationale and everything. So that's not to say that the report is less important on the FDA side, but again, I think that's really at the heart of the differences.
Okay. All right, I think we'll stop here. We're a little over 15 minutes over, so thank you, Jill. That's a wonderful presentation. And I think I'll turn things back over to Arlene to finish things up.
Yeah, let's close this out. Thank you everyone for joining us. Thank you, Jill and Kevin. You've definitely provided all of us good tips on how to implement a modeling and simulation program. We invite our audience to learn more about these approaches, and you can find out on our website. And you'll find today's presenters at the 2021 MIDD Plus Scientific Conference, March 3rd and 4th. This concludes our webinar for today. It has been recorded for playback and will be available on our website and YouTube channel. Have a great day, everyone.
Thank you.